For the coming presentation is Inhibition of intestinal tight junction-dependent leak pathway permeability by gallic acid as a cellular basis for the pharmacological effects of Ocimum sanctum flower aqueous extract, presented by Ms. Wanapas Wachiradejkul.
Good morning, everyone. Today, I would like to present my research about Ocimum sanctum flower extract, or OS-LF effect on tight junction disruption. In Thai, we call it Dog Kapao. But first of all, I would like to give you a brief background. Intestinal tight junction disruption is a key pathogenic factor that contributes to pathogenesis and disease progression, not only in intestinal but also systemic disease, shown in a list on the right-hand side. However, no drug targeting intestinal tight junction integrity is currently available in clinical practice. You can see tight junction in the epithelial cell. Once it is disrupted, antigen from the lumen flow through the paracellular space, triggering immune cell. These immune cells release cytokines which cause inflammation. Cytokines also stimulate myosin light chain kinase, or you can call it MLCK, resulting in tight junction-dependent pathway permeability. This will then become a vicious cycle as the antigen level rises, causing more cytokine release, and in severe case, if there's too much inflammation, it will lead to cell apoptosis. Once the tight junction-independent permeability stage is reached, ulcer erosion will have begun. It is clinically impossible to inhibit MLCK because it will produce systemically toxicity found in many systems throughout the body and have an effect on other parts such as smooth muscle paralysis. However, we also know that AMPK can increase tight junction reassembly and does not cause harm. Therefore, this is a better drug target to investigate our causes. In Thailand, we generally use OS-LF for herbs and for cooking, while the flower is a waste. But did you realize that? The flower cinder's core is filled with bioactive compound that can induce an AMPK-like action. And if the flower can induce lipophagy, tight junction integrity will increase. So our objective is to identify a major component within OS-LF that can increase tight junction reassembly and its underlying mechanisms. So to investigate tight junction integrity, we use T84 cells, our in vitro model. Transepithelial electrical resistance, or TR, was performed. Briefly, we placed T84 cell into our transwell plate. After that, we waited for 10 days for the cell to fully differentiate and form tight junction. Then we switched to calcium-free media to disrupt tight junction. And then 16 hours later, we switched back to calcium-containing media, and TR was performed. Consider a cell is aligned as a long sheet, and there's tight junction between each cell that prevents electrolyte or current to pass through. Therefore, tight junction acts as the resistor of the cell. So a high TR indicates a good tight junction integrity, whereas a low TR indicates a poor tight junction integrity. From our investigation shows that OS-LF increased TR in a time and dose-dependent manner. Next, we want to see the chemical component within OS-LF, so HPLC was performed. We then found that gallic acid is the major component. And to see if gallic acid increased TR in T84 cell monolayer in a time and dose-dependent manner and in a similar pattern as OS-LF. The result shows that gallic acid is the major component within OS-LF that can represent the effect of enhancing tight junction reassembly. However, TR does not fully represent the permeability pathway because TR drop could be due to either tight junction leak or apoptosis. Therefore, we need the permeability assay to identify the paracellular pathways. We insert a probe into the apical membrane, and if tight junction is intact, dextran cannot pass through. In the case of tight junction leak, only 4K Dalton can pass through. And in the case of apoptosis, both will be able to pass through. If dextran 4K Dalton can move through the basal chamber, we can collect samples from here and measure the fluorescent signals. So we use calcium-free media to induce tight junction, which shows an increase in 4K Dalton permeability, but not the 70K Dalton. This indicates the tight junction-dependent leak pathway permeability, and that both OS-LF and gallic acid can suppress tight junction-dependent leak pathway permeability. Next, we want to see OS-LF and gallic acid in intestinal epithelial cell viability, which shows a cytotoxic level. And this assay is based principally on viable cell being able to express mitochondrial reductase, which converts MTT to formosan and produce purple color. and this will not reoccur in dead cells. Our result shows that both OSLF and gallic acid doesn't have a cytotoxic effect. Now we know that gallic acid increases intracellular calcium, which was also known to stimulate via AMPK, SIRT1, and ERK. And this ERK pathway in particular was known to increase tight junction reassembly. Therefore, this is my hypothesis that gallic acid acts through this pathway. So we want to see if CAMKK is involved in these mechanisms. So CAMKK inhibitor was used, and we found that it can abolish OSLF and gallic acid intestinal tight junction reassembly. AMPK inhibitor was used and produced a similar effect as CAMKK inhibitor. SIRT1 inhibitor can also inhibit gallic acid effect and OSLF effect. Finally, we wish to see if ERK is also involved, and we found that ERK inhibitor can also disrupt the OSLF effect and gallic acid effect as well. So now we know that gallic acid increases intracellular calcium via CAMKK, AMPK, SIRT1, and ERK. Therefore, this correlates with my hypothesis. So we have successfully done in vitro. However, our findings should be further confirmed in a more complex model. And in the future, I hope that my tiny study could be turned into a medicine for the general population to treat disorders like inflammatory bowel disease or severe diarrhea, among many others. And this study demonstrates that it is achievable. Further benefit of this study not only shows that we can turn this agricultural waste into therapeutic drugs, but also help to boost the economic value. It has the potential to offer work possibilities for farmers, locals, and on a national level. Apart from improving healthcare quality, it could also help to build a sustainable agriculture. Finally, I want to thank my advisor, Dr. Pawin, for inspiring and encouraging me to become a medical student as a researcher, as well as Jelapang Royal Academy for their assistance. Thank you for your attention.
That's such an outstanding presentation.
Any comment from our judges?
Thank you for your presentations and nice work. From your HPLC result, that you try to separate the pure compound from your extract, right? Did you ever come across the second and the third major compound in your extract?
So when we send the OSLF to to test, we got back only the results for gallic acid because our aim at first was to find the major component within, and luckily we found that gallic acid can increase tight junction integrity, so we didn't send it back to find the second and the third compound.
All right.
Yes, sir.
Because in general, maybe I just give some suggestion because the major component doesn't mean or is not always equal to major potent compound. Maybe the compound that have small amount, maybe there is more potent than your gallic acid. But anything can happen. Yeah. So
in our experiment, we did many extracts, but just that the OSLF has the further development because there are also other extracts that we couldn't find the increased tight junction integrity. So we will have to do that as well to find the second or third until we find the one that can actually help. But luckily, we accidentally found that gallic acid is the major component, but also can increase tight junction reassembly as well. So it was more like a luck. Yeah.
All right. Because the data you show is very fast. Did you also perform some experiment with only inhibitors, so without gallic acid?
Yes, we did. Do you mean the mechanisms?
Yes, the TER experiment. Can you go back a little bit for the slide because it shows very fast, so I cannot follow.
Any more questions? Okay.
Can we switch to her presentation? Okay.
Could you please repeat the question again, sir?
No, I have no question. I just cannot follow this. It's a little bit too fast. Okay, then I have no further questions. Thank you.
Apart from the flowers, have you looked at the other parts of the plants? The roots, the trunk, the leaves. Have you compared the active agents?
Okay, so at first our aim is to find the agricultural waste in like different products that can increase like the health in humans. So at first we use the flower as a start to find the component that can help in terms of targeting drugs. But now we know that gallic acid has in flowers, so maybe our future studies could be to do with many other aspects of the Os Olaf itself. Yeah,
Yes,
but
yes,
because
good.
of the short amount of time, we didn't have time to do it in the leaves, the roots, but this is our further aim to do it as well. Yes, sir.
Okay, thank you so much.